Hello, my name's Andrew Dick and I'm the director of the UCL Institute of Ophthalmology in London, UK, and also chair and professor at Bristol Medical School in Ophthalmology in Bristol, UK. And today what I would like to do is take you through ocular immunity, some of its anatomy and repertoire, and in particular, try and be provocative to dispel some of the myths and talk about the wealth and depth and breadth of the ability of the eye to respond to immune challenges, to regulate its function in order to preserve vision. Right? The first interpretation which makes people think that the immune privileged site, first described by Peter Medawar, despite his Nobel Prize winning work, who displayed that corneal allografts can be accepted in a non-vascularized bed, is another bit of work where people noted that if they put protein into the eye, the front of the eye, as demonstrated here in the anterior chamber, it led to a systemic immunosuppression and a downregulation of delayed hypersensitivity responses when the animal was then challenged systemically. And this relies on the spleen. So splenectomized animals no longer show this response of immune deviation and immune privilege. But it's with this that people have noted the potency of the eye to immune regulate. And this has been termed anterior chamber associated immune deviation. Now the corollary is that it appears very much that we believe, therefore, that the eye is an immune privileged state. And indeed, it is an organ that can demonstrate extreme immune privilege and regulatory networks. But that's not the same as saying it's immune incompetent. So the eye is full of immune cells, as I think has been described previously in the talks. And I'd just like to highlight some of these again. The immune competent cells spread along the uvea of the eye, the iris, the ciliary body, and the choroid. They're present in the cornea in abundant numbers. They're present even in the retina. And particularly the retina is bestowed with a contiguous network of yolk-derived cells such as microglia. It doesn't have an anatomical lymphatic drainage, but it does drain to regional lymph nodes when the eye has been perturbed. And as I've said, it has very active immune regulatory networks, which I'll describe more about. So this is a light sheet of the posterior segment, the retina, just to demonstrate, because I like this video, of the network of the microglia in this reporter mouse to show you what the contiguous network is like. These are further on this microglia tomato reporter mouse that is yolk sac derived microglia. And as you can see, there are many of these immune competent tissue resident microglia, otherwise yolk sac derived macrophages, that are present within the tissue. And they're there to regulate immune responses. So jumping to the back of the eye, I put this schematic up to show several things. The first is the retinal pigment epithelium is, as we know, a non-canonical immune cell, but it expresses class 2. It can present antigen, albeit weakly. It has an immune regulatory function, mainly through its expression of Fas ligand, but other soluble factors. But it also orchestrates immune responses by secretion and activation of cytokines. Some of these interact with the choroid and the plasmacytoid dendritic cells and the myeloid dendritic cells that are present within choroid, other macrophages, and indeed mast cells, all which are present in the vascular layer underneath the retinal pigment epithelium. And indeed, internally, obviously, there's an interaction through cytokine and chemokine networks and cognate receptor ligand interactions with microglia that help maintain its regulation, but also can be perturbed to dysregulate to activate these cells to be very, very prominent immune response cells. Now, the reason of highlighting that was really to take a concept further. There's a concept called para-inflammation. I personally think this is a term that confuses, but actually it's a term that is also very meaningful. And what this is really demonstrating, for example, with age or with any insult, how the tissue can increase its inflammatory profile to regulate the tissue function and remove danger, and therefore restore back to homeostasis. Or in a chronic stage, it can become further dysregulated and become purely inflammatory. Now, this may be, as we see in this diagram, basal levels of tissue damage are maintained by the immune regulatory networks of the eye, maintaining homeostasis. As soon as we stress this, the tissue with disease, with therapy, or indeed germane to this meeting, with subretinal injections or intravitreal injections and viral transduction, we will induce a form of response in that tissue you'd expect. And it may be that it regulates under the para-inflammatory networks, or it can jump into and be pushed into more tissue injury and, as we would expect in any tissue, an inflammatory response. So I'd just like to break some myths and truths. When we look at inflammation, not all altered immune responses mean what we'd like to see as in acute inflammation, lots of cells. And I think the prima facie case here is systemic lupus erythematosus, where you get quite a profound antibody-driven response without undue inflammatory cells. You may get a little bit of neutrophil nets, but indeed when you biopsy, there's little evidence of acute inflammation otherwise. So many cells are not needed, and yet severe tissue destruction and attrition of function can continue. It is important to note that tissue regulation of immune responses in the eye remains as an active process. And certainly, I'm not going to go through all of this, but this again shows how the tissue itself is regulated by both chemokine and cytokine networks to regulate and indeed prevent often undue inflammation. So tissue function, when we're looking at regulation or we're looking at para-inflammation, can become attenuated when we reach a threshold of immune activation. And again, a prima facie case for this is in lung disease. So in lung disease, what we see very clearly is smoking continues to cause damage to the lung. It causes oxidative damage and other extracellular matrix changes. The resident macrophages are present, and normally they're alveolar macrophages, a bit like microglia, and they are there to regulate inflammation. But with smoking, their ability to do that and their threshold is altered. And as a result, the infections they would normally clear become more recurrent, more activated, such as in pulmonary obstructive airways disease. And what we see is an inability of these macrophages to regulate, and they then create chronic inflammation and tissue remodeling alongside frequent intercurrent infections. So another example to show the power of the eye and how it's dysregulated is, of course, a condition called uveitis. Uveitis is an umbrella term, and I'm just talking about the non-infectious types that are immune-mediated inflammatory diseases. They may be associated with systemic disease or not, but nevertheless, we believe that what happens is tolerance is broken to certain retinal antigens, as you can see, as normally projected for autoimmune disease. And with further innate stimuli, the dendritic cells are able to present this to T cells that classically expand, and as a result, they migrate and infiltrate the eye, bringing in other players such as the mononuclear cells and the monocytes and the neutrophils to cause damage in the eye. Now, uveitis isn't rare, it's not common, but it equally occurs round about 100 in 100,000 people. So the eye is very, very capable of generating inflammation. Similarly, sterile inflammation is a concept where we notice that any action not due to a live replicating virus or bacteria can induce inflammatory responses, normally through the inflammasome complex and caspase-1 activation. This could be induced through dying cells around them, could be induced from systemic features such as other areas of inflammation that can cause and infiltrate the eye with cytokines, or for example, with other oxidative damages such as smoking. What happens is we get an altered state within the eye to become hyperinflammatory or sterile inflammation. If we look at the retina during a low-grade inflammation, we can see quite readily that these cells are far more abundant than we can visualize by looking in. So I'm just going to play that again. So it doesn't look like there's many cells there, but when we look at the GFP expression, there are numerous amounts of CD4 T cells in the animal model of uveitis at a very, very early stage before we see really any clinical signs of inflammation. So we don't have to wait to see for cells clinically before we understand that this tissue may already be infiltrated and highly inflamed. So in general, when we look at the eye and its context of immune competency that normally controls the disease, it's because everyday stress in the eye has to be managed. We have to maintain a homeostatic state. We have a downregulatory immune environment, both at the RPE level, through microglia, and even when we look at the dendritic cells in the choroid, they tend to be tolerogenic. They have to maintain autophagy of the RPE and obviously normal function. But several things occur in life. One is the accumulation of oxidative stress and the sterile incitement of inflammasome activation that may occur, inciting an inflammatory response. We also have, as we see in aging and macular degeneration, complement dysregulation the older we get. And of course, that's even more emphasized depending on what genotype we have for certain complement genes. And then we have the concept of senescence and the senescence-induced secretory phenotype, again inflammatory. And again with aging, we have a loss of control of our regulatory networks, and systemically, we have an altered immune competency, often called inflammaging. Sorry about that term, but that's what it's called, where we're noticing with age, increasing age, we tend to become more inflammatory systemically. And indeed, the evidence we have from many tissues, including the eye, is exactly the same. So we lose an ability to regulate our immune responses with age, with accumulation of oxidative stress, with an inability to manage all these factors. And if we then lay on disease, albeit a non-inflammatory disease such as inherited retinopathies, but also by inadvertently potentially some of our therapies, it may be of no surprise that we may see inflammatory responses occurring. The problem we have, to finish, is that it's very hard to actually assess what's going on in the eye, either through imaging mechanisms at such high resolution to see inflammatory changes. And unlike cancer, we can't biopsy easily to ascertain exactly the mechanisms that go on. So I hope that's given an overview of some of the immune competency of the eye, particularly some of its anatomy, and most importantly, frankly, how easily it can be dysregulated. Thank you.